is a big commute. So we're going to start getting into the brain. We will get into exercise. I worked at a drug rehab clinic. I've mentioned this before in Connecticut for a few years. And there was somebody at the clinic who said, exercise. And it has lived on in my slides ever since. Exercise. So today it's mostly going to be talking about the brain itself, you know, how a brain functions. Everyone has one. That's what governs everything you do. It's what permits every action. It permits every thought. But we don't often think about the brain. I mean, it's just right there. And we don't often think about the meat itself conducting all of those verbs and thoughts in our lives. And so we're going to do a little bit of that. It's going to take a few lectures. Maybe we'll just run the brain and exercise through Thanksgiving. So make it 4 lectures, 3 or 4 lectures on this. At the end, we'll talk about exercise. We're not going to get to it today, but when we are a little bit closer to Thanksgiving, we'll be talking about the effect of physical activity and exertion and different types, different modes and intensities of exercise and how it affects the brain. But basic brain function, that's for today. You know your points, you know where to find stuff. Make sure this is always fundamental no matter who it is. So as we move forward, we'll have another case study, and it's going to be older adults. Right? It's going to be people aged, 60, 65, 70, 75, maybe 58 with a pathology. And what we've been talking about, senescence and these different expressions of cellular aging, because we don't really care. It's sort of arbitrary. Maybe we care, but it's arbitrary how many birthdays we've celebrated. That's not really what our odometer says. The odometer, just like cars, if you take care of a car, they don't all break down at the same time. If you take care of a car and change the oil and do whatever, they don't all exit the road and enter the mechanic shop at the same time. We don't care necessarily what the odometer says. A car can be destroyed with 4,000 miles on the odometer. Same thing with a body. Right? A body can be destroyed by the age of 40 depending on the inputs. And so age, cellular aging, how our body actually manifests the symptoms, the signs, the symptoms, the complications of aging, that's what we care about. And 50 and up, if there's a pathology involved, make sure you can figure out the functionality and prescribe accordingly. Now, remember, you can use alleles to predict disease states pretty effectively. Likelihood of X, Y, and Z conditions, cancers, or what. There are particular conditions that can be predicted pretty well with a set of alleles. But for athleticism, there's just so many spots that contribute the tiniest amount to human performance in whatever expression of performance that is. Resistance training, strength, speed, power, explosiveness, endurance, how far of an endurance sport, and injury susceptibility. All of those things are determined by so many different alleles. It's this huge constellation, right? It's not like the Little Dipper or whatever, or the big one, where you look up and they say, oh, there's 5 stars. That makes a spoon or whatever it is. It's the whole night sky of stars are determining an expression of athleticism, and whether it's VO2 max and so it's hard to find a single gene that says that's the sprinter's gene. Okay, alpha actinin 3 does a decent job. But is merely decent, and like half the world has it. The allele that's good, And so if you have the nonsense SNP, if you have that premature stop codon in alpha actin 3, you're not going to be a very good sprinter or power lifter. Fine. But that's just one star in the night sky. You need a ton of subjects in these studies to be able to predict with any amount of confidence the effect of particular alleles on athletic outcomes, and you need a good dependent variable. The dependent variable has to be some objective, measurable human performance assessment. And it can't just be on a scale of 1 to 10, how fit are you? Or were you pretty fast with shoes on? That's not good enough for human performance for the dependent variables. You need a huge sample and accurate assessments of human performance. And then those studies need to be replicated. And we often see failure of replication of a lot of these. Now, certain genes have emerged, certain alleles have emerged as very slightly predictive of particular expressions of athletic performance. Expression isn't the right word there because gene expression, but particular domains of athletic performance, some genes have emerged. But again, you can just go for a jog, and if you have good endurance, your genotype begets a phenotype that is predisposed to endurance sports. Go sprint, and if you outcompete your peers, you have a genotype, unless you've been training forever, you have a genotype that naturally permits phenotypic traits that would be consistent with sprinting performance. Okay, the brain and exercise, but we're not going to talk about exercise yet. Exercise is where we arrive. That's the finish line, is exercise. But we're going to talk about the brain a little bit first. So maybe we can do it in 3 lectures, but I don't think so. I think it's probably going to take us 4. So the 4 lectures covering these 3 topics: basic brain function, what the brain is, what it does, and how it does that stuff. That's where we are today. Then we'll talk about the ideal performance state, what people would call the zone or flow states. You see this in sports, but you see it everywhere. You can get it with writing. You can get it with writing with a D also. You can get it with any sport. You can get it with video games. You can get whatever it is that you do where you get that perfect focus, where it feels like you can make no mistake. You all know this state, right? But again, it's commonly associated with sports. People get it in music all the time. These are some of the most, the experiences that define our interests. Wherever you're able to experience these ideal performance states, rock climbing or whatever, wherever you're able to experience those states, that says something about what you should be doing in life, what your real interests are, not what you want to do. I always wanted to speak a bunch of languages, but not really, because I'm horrible at learning other languages. I did Japanese for years, in high school and in college, my undergrad. I did Japanese, and I'm horrible at it. I tried to learn French for a while, and the only thing I can remember is "Où est la rue Saint-Jacques?" which is like, where is St. Jack Street? But I've never been somewhere where I could ask that question, and St. Jack Street was a real place, was like a non-fiction place. And so, but I tried and tried. And then I realized what I wanted to be the kind of person who wanted to learn languages. Same thing with people who read types of literature that are inconsistent with their interests. I think high schoolers should not be reading, F. Scott Fitzgerald. And I don't think they should be reading that stuff. I think they should be reading like Harry Potter. Stuff that they like, stuff that they connect with. Not stuff that they think they should connect with. Oh, I want to be the kind of person who connects with this. I want to be the kind of, have socio-political commentary at that, you're not going to achieve an ideal performance state pursuing something that doesn't connect with you. And so we'll talk about the types of environments that predispose you to that, and the sort of neurochemical cocktail that elicits it. What's going on in the brain that achieves this? And some strategies of how to make it happen. And then the last thing is going to be brain health and exercise. That's going to be where we end with all this. But for today, the basic brain function. So here's our brain. We have, our brains, plural, there's one of them. And for a long time, people said, you have, this 100 billion neurons in your brain. That's the expression. We have this 100 billion neurons and way more glial cells. But when you try to figure out where that number came from, I mean, do you know how long it would take to count 1, 2, 3, 4, 5 to 100 billion? So how are people getting this number? The number's reported over and over and over. And so this study in 2009, they tried to estimate somewhere close to the real number of neurons in glial cells or non-neuronal cells in the brain. And for the whole brain, 1,500 grams, 454 grams per pound, right? It's a pretty light organ, and yet it consumes about 1/5 of our energy. That's about 20% of our energy and a couple of percent of your body weight. So it's pretty light, but the number of total neurons, about 86 billion, give or take 8 billion. That's the estimation that they came up with. But when you look at the cerebral cortex, this huge hunk of brain meat, right? This huge, it's about 82% of brain mass, but only about 19% of the neurons. Compare that to the cerebellum back here. It's about 1/10 of your brain mass and about 80% of your neurons. So about 1/10 of the mass, about 10% of your brain mass, and 4/5 of your neurons live in the cerebellum. So this is a neuronally dense space back there. And then the whole cerebral cortex, it's not that neuronal compared to the cerebellum. But we have, it's fine to round up to 100 billion, but somewhere between maybe 80 and 100 billion neurons, something like that. There's a lot of these. And what is a neuron? In Travis Stiles' lecture, Kevin asked a question about the graft. There was a neuronal graft. And he was like, it's not the nerve, it's the tissue. Really, this is tissue over here. This axon isn't the nerve. The nerve, here's the nucleus, the cell body of the nerve. That's the nerve. Everything else is just sort of tissue. But the nerve, what we count it as really is a cell body. We have our dendrites. So let's receive input, let's receive input, let's receive input, let's receive input. Maybe it's going to receive input from like 100,000 sources and then discharge its sauce. So it goes down the axon and releases from the nerve terminals. It's going to release maybe dopamine, maybe acetylcholine, glutamate, serotonin, any of these neurotransmitters can be released from those nerve terminals to communicate with another nerve, potentially, probably another nerve, or some tissue, some organ it can communicate with. So if this were a skeletal muscle over here, and this were a lower motor nerve, upper motor nerves, right, live in your bones, inside of your bones, right, inside of your spine, inside of your skull. Those are upper motor nerves, central nervous system stuff. Out in the peripheral nervous system, we have lower motor nerves. And it's synapses at the spine, and then it reaches out toward the tissue, toward the muscle. And that long axon, and then we have our neuromuscular junction, right, that synapse, where the nerve merely touches the muscle. But normally what we're thinking of is one nerve ends, another nerve begins. There's a bunch of dendrites over here too, picking up those signals. Now the glial cells, the astrocytes are one type of glial cell. They are critical for the nerve's function. And then the oligodendrocytes, that's another type of glial cell. So these glial cells are critical for the nerve function, but they're not transmitting messages. Nerves are what are transmitting the messages. Nerves are releasing those chemicals, those neurotransmitters, those chemicals, the dopamines and acetylcholines and all of that. That's being released by nerves. These are sort of, you can think of as support staff, these glial cells. Now, in the developing brain, so this is from the National Institutes of Health, right, NIH.gov. So in a developing brain, the neuron depends on signals from those glial cells. So our development of the brain. Now, once we're born, once you're a living human being with open eyes and you see something other than a womb, classically, what was always said was that you have the number of neurons that you will have, that's your peak, right? And like watch TV or drink alcohol and you're killing brain cells and brain cells never grow back. That is what has always been said. And we just decay throughout life. And it's tough to regrow nerves. Travis talked about some of these mechanisms, ROA, right? The inhibition of neuronal growth. It does happen, and we'll talk about how it happens. It doesn't happen abundantly. It doesn't happen abundantly, but it does happen. And in lab, like Fred Gage was a researcher who's in his lab. He demonstrated it in human beings. It's been demonstrated in tons of it in rats. Particularly looking at areas like the hippocampus. We'll get there. We'll get to that stuff. But a lot of these cells are determining or helping. They're the molecular signals being provided that help determine the development of those neurons. So again, here's a nerve, right? The nucleus, the cell body with the nucleus in it. The dendrites are receiving inputs, signals are being received over here, and then we send that message down the axon to another nerve, right? The postsynaptic cell, the postsynaptic nerve, these dendrites. So the terminals from this nerve release their neurotransmitters to stimulate, to excite, to inform the next nerve. And these little things, this is myelin over here, the myelin sheaths. And it's called saltatory conduction, where it just leaps, right? It just, in the way that the post office works. Put tracking on an envelope or package and follow it. And it goes from Stockton, and then next stop, immediately it's a Stockton. And then it doesn't say like, we're on I-5. We're traveling on I-5, there goes Lodi. It doesn't do that, it's a Stockton. And then Sacramento, and then they'll say like Utah, or something, and then like Nebraska somewhere, like York, Nebraska, it's like, whatever it is, that's like saltatory conduction, where it just leaps, depending on the thickness of that myelin, it'll go faster or slower. Remember when we were talking about pain nerves, nociceptors, and the A deltas are thinly myelinated. These little myelin sheaths are pretty thin, and so it conducts, from beginning to end at, Olympic sprinter speed, where C fibers that aren't myelinated are me pacing in front of the classroom. That's your C nociceptors, pain fibers. But, afferents, we have afferents, we have sensory nerves. And for those sensory nerves, we also have A alpha and A beta. And those are thickly myelinated, and those are like racing around Lamborghinis. But so that would be nerve to nerve conduction. And there are a few kinds of nerves. So this is a muscle spindle. When you go to like, an athletic trainer or a doctor or somebody, and they do this thing, and you kick. That's the muscle spindle response. It's a stretch flex response. And like everything, it is in the interest of self-preservation. If a tissue is being stretched and that stretch is not obstructed or inhibited or somehow halted at some point, you're going to stretch to the point of injury. You're going to disrupt tissues, you're going to hurt yourself. And so you have this stretch flex response. And so that's the muscle spindle response. So that's all we're talking about here. But there are sensory nerves, right? So we have these sensory nerves that come in the dorsal root, right? So afferents enter the spine in the back, the dorsal, like a dorsal fin, right? It's all up in the back. The dorsal root, that's where sensory nerves enter. Motor nerves exit the ventral, the front. Motor nerves enter the front. They exit the front, aka ventral. But then we have interneurons as well. There are classifications of these nerves within these categories. So it's not like, well, there's one kind of sensory nerve, right? There's one kind of motor nerve, but that's not true. But even here, we're looking at a couple of different potentially motor nerves. The gamma motor nerve is what activates the muscle spindles, and alpha motor nerve is voluntary. So there's different types, these are the categories of our nerves, right? We have sensory, afferents, right? We have motor, efferents. And then we have interneurons. So what an interneuron is doing is, like with the muscle spindles, we get this stretch information, including load and rate of stretch. Like how forceful is this stretch? And the tissues contract accordingly. You know, the reason you go down before you go up, the reason you wind up before you throw, stretch, flex, response. And it comes up to the spine, enters dorsal root, and then informs an interneuron. And that interneuron informs the gamma motor nerve. Now, if this were a Golgi tendon organ, right, which says that's too heavy, relax. You're going to hurt yourself if you keep contracting, so stop contracting, relax. Again, you have sensory nerves, and then you have an interneuron. So all this activity comes from the spine. This doesn't go up to the brain. The brain isn't making these decisions. You don't consciously decide to kick from the brain. The interneuron informs the motor nerve what to do. And with a Golgi tendon organ, it's actually hyperpolarizing. It's glycine. We'll talk about that later. You're hyperpolarizing an alpha motor nerve to inhibit it. So interneurons, these are in the spine, right? This is a reflex arc. You're in the spine. This is not brain controlled. Cut off your head and this still happens. This isn't coming from your brain. And so the brain is the primary area for these neurons to be controlled, but they're not all controlled in the brain. You have spinal control of these things too. Now the synapse, that nerve to nerve conduction or nerve to tissue conduction, if you've been on a car ride with a sibling and you have a fondness, loathing relationship with that sibling. It's a long car ride and like your dad's driving and he has a thing about not pulling over until you get there. So just hold it or piss your pants or whatever. Put in a catheter, but I'm 5. We'll wear a diaper then, I don't know, we're driving 9 hours. So if you have had this experience where you're sitting in the back of a bumpy seat and you have 9 hours to entertain and your sibling is right there playing Game Boy Advance or something, and you go, I'm not touching you, I'm not touching you, I'm not touching you. But it's like you've penetrated my aura, right? My, I don't know, my chi extends that far out of my body and you punctured it. That sort of proximity, where it's basically touching for practical purposes, for emotional purposes, for purposes of maintaining composure, of not becoming frustrated. It's touching. But that's what these nerves do. These terminals, so she's like a dendrite, or so she's like muscle tissue or something. And he's this terminal, this nerve terminal saying, I'm not touching you, I'm not touching you, I'm not touching you. But there's this tiny, literally the letter of the law, he is not touching. But the spirit of the law might as well be, but there is a chemical communication that the nerve releases whatever it is. Let's say it's muscle and it gets acetylcholine is released. Now acetylcholine has muscarinic receptors and nicotinic receptors. It depends where you are in the body what acetylcholine is doing. In skeletal muscle, it's nicotinic receptors. So it's a little bit different. Now, long-term potentiation, right? This is, let's strengthen the relationship between one neuron and the next. Long-term potentiation. Long-term depression would be, let's weaken the relationship. But long-term potentiation, this is learning, this is recall, this is when you're studying for a test and you're doing the flashcards over and over. Repeat exposures, right? You're having these you see the question, you see the answer, you see the question, you see the answer. You're trying to strengthen that connection until it's strong enough to recall on your own. When you're taking free throw after free throw after free throw, whatever it is that you're doing repeatedly, you're practicing scales, right? Or on guitar or whatever, you're practicing scales. What you're doing is trying to establish long-term potentiation, trying to establish stronger relationships between these nerves, between neurons, between some particular set of neurons. So long-term potentiation and long-term depression would be the inverse of that. It's a weakening of those relationships. Now, most neurotransmission, most of this chemical communication in the central nervous system. So let's get up in the brain and have our central nervous system communicating nerve to nerve. The primary excitatory neurotransmitter, what nerves are releasing to excite, is glutamate. The primary inhibitory neurotransmitter saying, no, calm down, calm your ass down over there, is GABA. You make GABA from glutamate. But the primary excitatory in the central nervous system, the primary excitatory neurotransmitter is glutamate. The primary inhibitory is GABA, gamma aminobutyric acid, GABA. Now, glutamine to glutamate to GABA. So we all know what glutamine is, but you go from glutamine and glutamate back and forth. Astrocytes, you can do that. And then there's a mitochondrial enzyme convert back to glutamate. And so primary excitatory, primary inhibitory neurotransmitter. Monosodium glutamate, it's pretty much the same thing. You know what a proton is, right? So the electron's a proton, but a hydrogen ion is just a proton. And we'll talk about that when we're going through glycolysis of pH, H, hydrogen ions, right? Proton. So if it's protonated, you're throwing on this hydrogen here. But so here's glutamate, and then protonated and put on a sodium. There's MSG. So when you go to like the Chinese buffet, and there's that flavor, we all know what MSG is, right? That's glutamate with a little sodium in it. So that's all that is. Now, when we're looking at learning, now learning could be mathematics, learning could be the piano, learning could be how to do a power clean. Learning is, maybe it's motor mapping, or maybe it's memorization, how many digits of pi. Learning takes on a lot of forms. And this is a study that was looking at MA, you see up here, MA. That's not Master of Arts, that's mathematical achievement. And what this study, this isn't every study, but this is a good study on glutamate and GABA and those relationships with learning. And what they found was early in life, I mean, really young, those formative years, GABA may help assist in learning. Later years, all of you, everybody who would be old enough to listen to this, glutamate is your learning neurotransmitter, and GABA is inhibitory to that. Very early in life, it could be reversed, sort of setting stages. So just to read their findings. Furthermore, we were able to extend our findings and show the inverse effects for, what the hell was IPS? for mathematics and whatever, for GABA. Mature individuals showed a negative association between GABA concentrations and mathematical achievement, while younger participants showed a positive association, and then the inverse. So glutamate and GABA, certainly for adults trying to learn, it seems to be a glutamate function. But again, learning is, there's so many domains of learning, and sometimes GABA can be of value, right? Depending on the nature of learning. Some calm environment, some non-anxious calm environment could be contributing to focus and learning. So it gets a little bit interactive or complicated depending on the nature of learning. But NIH, again, glutamate, the most common neurotransmitter in a person's body, which increases neuronal activity, is involved in early brain development and may also assist in learning and memory. The literature is a little bit scattered, but that's a good summary statement. Glutamate, most common neurotransmitter, it's an excitatory neurotransmitter, and it is critical to learning. So there are different receptors for glutamate. You know, there are receptors for everything. You know, we went through mTOR. You know, you have your IL-2 receptor, you have your IGF receptor, you have your insulin receptor, you have androgen receptors, you have TNF receptors, you have receptors for everything. Glutamate, there are particular receptors for too. Cell surface receptors, sure, on a nerve cell surface. And so there's ionotropic glutamate receptors, and then there's metabotropic glutamate receptors. You're very familiar, even if it doesn't sound like you are, you're very familiar with metabotropic receptors because it's cell signaling. Cell signaling cascade, metabotropic receptors, second messengers, just that cell signaling. Ionotropic is ions, positively, negatively charged things, stuff with a charge, right? Let's let that through. Ionotropic receptors. So this is a postsynaptic neuron. And there's glutamate and glycine. Here's glycine on the NMDA receptor. It's the only place you see it. So glycine is an inhibitory neurotransmitter. Glycine is an inhibitory neurotransmitter. If you can inhibit glycine, you can improve performance a little bit. Strychnine, like poison. Strychnine is a competitive inhibitor of glycine. That's the reason strychnine, before the advent of science and sort of chemists entering the ergogenic aid workforce, strychnine was a really popular ergogenic aid in sports like cycling. Dangerous, sure. Potentially lethal, sure. But it is an inhibitor, a competitive inhibitor, binds where the other things was of glycine. But glycine has another effect. Glycine assists, sort of co-agonist for glutamate's NMDA receptor. So glycine has a couple of effects in the body. But ignore glycine and focus on glutamate. Here's a presynaptic neuron. We're releasing a bunch of glutamate. These little vesicles, right? They're gonna come down, exocytosis, let's dump our stuff. Let's dump our stuff into that cleft right here, the synaptic cleft, the synapse, right into this space. Again, if this were a muscle right here, this would be, let's dump some acetylcholine out and bind to those acetylcholine receptors on that motor end plate, on the sarcolemma, the other side. But this is a nerve. This is a nerve, and that's glutamate, an excitatory neurotransmitter. So the way you dump this stuff, there's like the kiss and run, it's sometimes called, where you come down, you drop a little bit, just a tiny bit, and move on, right? You just a little touch. And then there's a partial release, or open and close, and then there's a full release. So it's not as though every single circumstance results in a complete dumping of all of your sauce, right? Your glutamate juice. You can control how much is being released. Not consciously, you know, okay, let me just control how much of it, but your body will not, it's not like an all or none principle. You know what determines it? You know, the excitement, the excitatory stimulus, but do I know what determines it from the perspective of the mechanics? I don't. But the you know, degree of the excitatory stimulus, maybe there's inhibition somewhere. You can take drugs that prevent the release of things. And so the mechanics of it, I don't know the answer. So looking at the postsynaptic neuron, this glutamate is binding to all of this stuff. What are these things, right? So it's released from the terminals, crosses the synaptic cleft, binds to that stuff. The AMPA receptor is letting in sodium. Sodium's positively charged. If you let in a positively charged thing, what happens to a nerve? Depolarizing, right? So AMPA receptors, let's let in some sodium. It's mostly acute state, not entirely, but mostly this is acute effects on the nerve. NMDA receptor, this is the one glycine is you know, playing a role. This lets in both sodium and calcium. Sodium and calcium enter the nerve. Calcium has so many regulatory functions. So NMDA activity can help determine the genetic expression of receptor concentrations. You know, how many of these guys do you have? You know, is this just loaded with these receptors? Getting a bunch of calcium into the cell can influence that stuff. So when you're looking at long-term potentiation, when you're looking at long-term depression, those NMDA receptors become important. These ionotropic receptors. Let's allow ions to pass through. What's called kainate receptors, these seem to be less important. They're certainly less understood, but they seem to be a lot less important also. So the AMPA and NMDA, definitely remember those ones. And then the, but again, increasing the number of these receptors. If you have, like for skeletal muscle, let's say, let's just keep going back to that analogy. Myasthenia gravis, literally grave muscle disease, is an autoimmune disease in which you break down your acetylcholine receptors, right? On the motor endpoint, you break down your acetylcholine receptors. And it doesn't matter how much acetylcholine gets dumped into that neuromuscular junction. If there are fewer acetylcholine receptors, you get a weak response. And so that's why you see myasthenia gravis having like ptosis, right? This sort of drooping eyelid. The motor activity is compromised. That same phenomenon happens no matter what receptor it is. If you downregulate the receptors, you diminish the input, the signal, whether it's motor activity, neuronal activity. And so long-term potentiation, let's increase the same dose of glutamate or acetylcholine or norepinephrine, whatever it is, the same dose would elicit a more pronounced effect if you have more receptors. The same dose would elicit a diminished effect, long-term depression, if you have fewer receptors. Now remember tract A, nerve growth factor, binds to tract A, tropomyosin receptor kinase A. This is a metabotropic signal. That's a metabotropic signal. It's a cell signaling cascade. And we have glutamate metabotropic receptors too, that initiate signaling cascades. Second messenger systems regulating postsynaptic protein synthesis. Remember, proteins do everything in the body. Receptors, those are proteins. All this stuff is just a bunch of proteins. This is a slide from our SEPA lecture, that brief SEPA lecture. There's track A, there's track B, probably. I can't read anything. But BDNF, brain-derived neurotrophic factor, BDNF, is that binds to tract B, and we'll talk all about that on next week. We'll talk all about BDNF. Nerve growth factor binds to tract A, tropomyosin receptor kinase A, or receptor tyrosine kinase, so that class, and does a signal transduction pathway. Now, one of the functions of these metabotropic receptors, there are a few classes of metabotropic glutamate metabotropic receptors, and one of them inhibits adenylate cyclase. So if you inhibit adenylate cyclase, you inhibit that signaling, and you know what that signaling looks like in a fat cell. You know what, like epi or norepi binds, activated adenylate cyclase, that breaks down ATP into cyclic AMP, cyclic AMP binds to PKA, PKA phosphorylates tons of stuff. If you're in fat, you're phosphorylating pyrrolipine, you're phosphorylating hormone-sensitive lipase. If you're in the liver, right, you're phosphorylating phosphorylase kinase, which phosphorylates glycogen phosphorylase. Glycogen synthase, you're phosphorylating different things depending on where you are. If you're in a nerve, right, there's a different phosphorylation cascade. But, so that's that one right here. Inhibit adenylate cyclase, shutting down cyclic AMP signaling is that second messenger. Another class, right, you're increasing calcium concentration. Another class is the release of potassium, and then more calcium. So there's a bunch of calcium, potassium, and some cyclic AMP. So these metabotropic receptors are cell signaling cascades, and the glutamate ones have several functions. Now, there's a cliche which is descriptive of this phenomenon, which is what fires together wires together. I hate cliches, but that one does characterize this phenomenon pretty well. And neuroplasticity is a constant process. Neuroplasticity, the changing of your neuronal structures is forever ongoing, right? It's not like, well, when you were a kid, you had neuroplasticity, and then you're not a kid, so you don't have it. It doesn't work like that. Now, not all neuroplasticity is good. You know, people who have trauma in life, we rewire. Like people who, if you were born and raised in Stockton, you're more likely to lock your doors all the time. Okay, where I grew up, we sort of like don't lock your doors. And so in Stockton, I've been conditioned to like, I don't need to lock my door. But trauma, that's not really like serious trauma. Trauma is like some form of abuse or some, like a diagnosis of some kind. Neuroplasticity can be very pronounced and rapid in those situations, or neuroplasticity can be very good. The idea is we want to create an environment, to create a social structure, an exercise environment that facilitates optimal plasticity, optimal neuronal health, that permits better functioning. So Eric Kandel, this guy, he's still a professor. I looked him up this morning. This morning, I went to columbiapsychiatry.org. I don't know why it's .org. But he's apparently at Columbia. He's still a professor, even though he was born in 1929. But he won a Nobel Prize for figuring out how these synapses, this nerve-to-nerve connection, how it gets strengthened, on repeat firing. Remember, fires together, wires together. That stupid rhyme, but it's very descriptive. So when these nerves, when you take 50 free throws in a row, and your body learns how to do it. When you take, you go to the batting cages, and you just swing after swing after swing, and you start mapping motor pathways. When you practice your chromatic scale, Kevin's not here to talk about music, but when you practice your scales over and over and over, interestingly, a lot of this plasticity happens while you sleep. We're not going to talk about sleep in this class because I like OD'd on sleep in stress phys. For those of you who are in it, remember that 5 straight lectures of nothing but sleep talk. But when we sleep, if you practice your scale, practice your scale, practice your scale, and you get to this point where like, you didn't do it, and you're frustrated when you go to sleep, you wake up the next day, and the scale's fine. Your fingers just do it. Or maybe it's on the piano or something. And that doesn't look like a scale on a piano though. And so a lot of the plasticity, actually, the follow-up alterations happen while we sleep. But there's a lot of overlapping and intersecting and contradicting signaling in a nerve. So like look at a pain nerve. Well, here's PKA signaling. There's adenylyl cyclase and prostaglandins. Right? Prostaglandins are, like let's say you have exercised and you're having delayed onset muscle soreness. Prostaglandins are a arachidonic acid derivative leading that charge. And let's turn on adenylyl cyclase and accumulate some cyclic AMP and get some PKA signaling. Well, what if we do weed, right, or opiates? What if we do those? We're inhibiting adenylyl cyclase signaling. So there are these interacting and inhibiting compounds. Substance P, bradykinins, there's prostaglandins again, histamine, 5-HT, that's serotonin right there. CGRP, calcitonin gene-related peptides. So substance P and CGRP, the nerve itself releases. The nerve releases those, and then it stimulates the nerve itself. So the nerve releases stuff to stimulate them. Oh, glycine and protons, the pH. AMPA receptor, NMDA receptor, all of this stuff regulating the function of a nerve. But there's parallel roads, right? There's intersecting roads. It gets busy inside of these nerves. We'll focus on glutamate though. So tell me what glutamate is. The primary excitatory neurotransmitter. What kinds of receptors do we have? Yeah, so ionotropic and metabotropic. Metabotropic, we have cell signaling cascades. Ionotropic, let's permit some ions to go through. Which ionotropic receptor does just sodium?
AMPA.
AMPA. Which one also lets calcium in?
NMDA.
NMDA. And okay, we can just ignore those ones. What do you make GABA from? Glutamate. So let's get beyond glutamate and look at some other neurotransmitters. We'll do a little bit more justice to neurotransmitters as we go. This is just a very cursory surface overview of some of these other neurotransmitters. And we could put, there's a ton more, and we'll talk about some of these other ones. We've been talking about acetylcholine a lot today because of the nicotinic receptors, because of skeletal muscle. But during some sort of intense psychological phenomenon, you're going to get acetylcholine as well. And it has a different function of some of these other ones. Norepi, this is let's focus, let's be alert. We're vigilant and narrowly focused and hyper attentive, right? Norepinephrine. But you're going to get acetylcholine with that. You're going to get acetylcholine with it. And what acetylcholine helps do in those situations is identify the neuronal experience, identify qualities, and sort of shine a light on them. Say, this needs to be, we need to remember this. We need to wire this together, right? We need to pay more attention to this. So that's really what acetylcholine is doing in some of these. It could be you're running with the bulls. It could be somebody broke into your house and you're in it. It could be like you're almost in a car wreck. It could be a stressful, emotional relationship. Any of these things that potentially shorten telomeres. Any of these emotional, psychological situations norepinephrine, oh man, you're gonna focus. I mean, tunnel vision on that thing and hypervigilance. And then that acetylcholine is going to help identify, kind of highlights, it's shining the spotlight on it. But what a lot of these things are doing, serotonin, norepinephrine, dopamine, we'll talk about those three today. And we'll get into stuff like anandamide later. Some of these other compounds we'll talk about later. But instructing neurons to make more glutamate, that's one function that these would be doing. Instructing the neuron to make more glutamate. Altering the sensitivity of the neuron's receptors. What's type 2 diabetes? Tends to get later on in life. It's a little bit, in today's.
Desensitized your insulin.
Desensitized insulin receptors. Yeah, so receptor sensitivity is a thing, right? And so altering the sensitivity of a neuron's receptors, that's not insulin receptors, but you can think of the phenomenon as similar. Make neurons more efficient. So what that means, what some of these other neurotransmitters are doing, making neurons more efficient. What that means is think about it from the perspective of exercise. You've never exercised before. And now I want you to run a mile and then bench press 100 pounds 20 times. And then walk half a mile with lunges. Every step is a lunge step. Go do that. If you've never exercised before, it takes the week out of you. Like you're in bed. If you can finish it at all. And then you get better and better and better. Eventually, you would get so good at this, you would adapt. You'd be so efficient at it that you wouldn't even be sore, right? That you wouldn't even be tapping into these sort of reservoirs of effort. In matters of like type 1, type 2 motor units, you can start to accomplish what used to be a type 2 task, a high threshold motor unit task with type 1s. Or fewer and fewer motor nerves, or depleting energy resources less and less and less. So that's the idea of this efficiency. You can see it when in sort of academic training, it takes less cognitive work to accomplish the same task. Deliver signals directly, right? These have their own receptors, so you can be delivering the signals directly, not just augmenting glutamate, or affecting the signal to noise ratio of these things. So serotonin, we'll talk more about serotonin, especially as we get into exercise. We'll talk more about these things. Again, very cursory introduction to these, but it's sort of regulating mood. And when you look at SSRIs, the selective serotonin reuptake inhibitors, mood is what a lot of people will talk about. Impulsivity, anger, aggressiveness, ignoring all the bowel stuff. So serotonin has a lot of regulation with that. And then also, this is from a 2005 study that just has one kind of nice line in it. Although it can generally be said that serotonin enhances the production of dopamine and hampers the production of cortisol. So there are relationships between these. It's not like serotonin does its thing and then dopamine's over there doing its thing, and there's no interaction. There are interactions between some of these things. Norepinephrine, again, this is your Marshall stack. This is like, you turn it up as an amplifier. You turn it up, and this captures your attention. Norepi, remember epi and norepi, same thing as adrenaline and noradrenaline. Root words, who remembers the root words?
On top of the kidney.
Yeah, adrenal, INE. INE is hormone, and on top of renal, the kidney. What about epinephrine? Same. Same. Epi, like an epidemic. Atop the nephrologist, kidney doctor, nephrologist. INE, epinephrine. So hormone from atop the kidney. Noradrenaline and norepinephrine, these are the neurotransmitter version. The nor version is the neurotransmitter version, as opposed to the epi or adrenaline, which is the the endocrine version. Let's release it from the adrenal glands, the adrenal medulla or medulla. So amplifying, that's norepi. Now dopamine is interesting. All of them are interesting, but dopamine, there's a lot of reward and trying to decipher the algorithm that dopamine has. So, you know, a lot of satisfaction and reward. And so slot machines, when you look at gambling studies, dopamine is a huge target of gambling studies. And so mesolimbic dopamine, so this is what regulates, what motivates gambling behavior, insight into dopamine's role. DA, mesolimbic dopamine, chief neuromediator of incentive motivation, is indeed released to a larger extent in pathological gamblers than in healthy controls during gambling, as in other forms of compulsive or addictive behavior. A little bit of excess dopamine, and you're going to be hitting that slot machine more frequently and for longer hours and more bankruptingly. So in pathological gamblers and healthy controls, dopamine release seems to reflect the unpredictability of reward rather than the reward per se. You know, the reward on its own, right? The reward on its own is not necessarily the dopamine, it's the unpredictable nature that you're trying to decipher. And you don't even really need to get too far into this one. All of these articles will be on the website. But dopamine release in ventral striatum of pathological gamblers losing money. Multiple studies, this is a normal finding. This isn't like some weird thing, this is a normal finding. On loss, when you pull the slot and you lose, pathological gamblers get a dopamine surge. That's probably one of the primary determinants of what draws somebody into pathological gambling, is trying to win back those losses, trying to figure out the algorithm. No, the cherry came first 3 times ago, and then what I saw were the 7s. And so on my next one, it's random. This stuff is random. Your dopamine, your nerves don't know that it's random, right? You might consciously understand that there is no algorithm, there is no pattern, there's nothing to be deciphered here. But your dopamine has no idea. And so it's constantly trying to figure this out and give you little surges. And in pathological gamblers, you see this augmented dopamine signaling with the loss. The win, it's about the same, but with the loss. Now, dopamine is another, when you look at literature, and I'm wearing a shirt from one of my favorite writers. Does anybody know who Pat Rothfuss is? So he wrote, it's not this shirt, it's the one underneath. This is a very, this is my version of a striptease. So you have died, Oregon Trail. Like, you have died of dysentery, and that's always that. So the Chandrian are this, it's like a mystery. You're trying to figure out who the Chandrian are in this book. So you have died of Chandrian. And so they're these, you don't know, they're probably the bad guys. But what it's Name of the Wind is the book, and then Wise Man's Fear is a sequel. It seems like 1,000 pages each. And I went through them beginning to end twice, both of them. And it's the dopamine, where you're trying to figure out what the riddle is, and what people used to get in like crime, and you're trying to solve, it's the whodunit stuff, the Sherlock Holmes. And what keeps you hooked is there is an answer, there is a riddle to be solved, there's something to decipher, and you're trying to put together the clues, you're trying to do the deciphering. And this works really well in literature. I mean, just ask like Dan Brown or something, the Da Vinci Code guy. I read that too. That's not one I would read a second time. But this dopamine that we're going to get from trying to puzzle together the scrambled pieces. Now, most drugs, most pharmacological agents are targets. They're targeting serotonin, they're targeting dopamine, sometimes GABA. And Prozac, this is figuratively, but as literally as it gets, the mother of SSRIs. It was the first antidepressant drug, was Prozac, the trade name of Prozac. And SSRI, remember, selective serotonin reuptake inhibitor. And where you would normally reuptake the serotonin, you lock the doors and say, nope, keep playing in the street. It's like you're a parent and your kid needs to get more physical activity and they want to come in and watch TV or play video games or something, and they like lock the door and no, just keep playing in the yard. All right, I'll keep playing in the yard. And so that's the effect of Prozac in terms of its antidepressant effects. Anti-anxiety commonly targets GABA, primary inhibitory neurotransmitter. We're commonly targeting GABA in anti-anxiety medications. But in the last lecture, a couple lectures, at some point recently, I mentioned that there was a study. Kevin asked a question, and I don't remember what the question was. It was something about, I have no idea what he asked, but I talked about this exact article right here. It's from New England Journal of Medicine, and selective publication of antidepressant trials and its influence on apparent efficacy. So that's New England Journal of Medicine, top journal in the world. And then John Ioannidis, published one in the same year, 2008, looking at this sort of the myths of antidepressant effectiveness. And so it was a telomere question. I can't remember what it was. It was telomerase and antidepressant medication or something like that.
I think it had to deal with how exercise can be an inflammatory response, but then it's like that telomeres.
I remember that question. And that one, I sort of had an easy answer queued up for. I think I just talked about the heart rate and stuff. But he asked one that was about like the depression and antidepressants and telomeres or something. And I said, it gets a little bit complicated because we don't even really know the efficacy of these things. And you do see potentially an increase in telomerase activity with antidepressants, but they don't publish all of them. And so both of these journals, but both those articles will be in the file folder, and they're both really good. Talking about the publication bias of the pharmacological industry, pharmaceutical industry, and the consequences of that are assuming that they're really effective. Now, where we're going to go next class, we'll end here, but where we'll go next class is getting beyond the neurotransmitters and looking at a bunch of proteins, factors. A factor is just a protein, right? Insulin-like growth factor, that's just a protein. Vascular endothelial growth factor, fibroblast growth factor, brain-derived neurotrophic factor. All these factors, these proteins, that help regulate a lot of the cellular activity. So we will end there today and pick up there on next week. Are we okay with neurotransmitters and brain health and brain function and whatever?